Hi everybody, it is my very great pleasure to welcome you to this ALK event here this evening in association with Lung Cancer Europe, which is LuCE, and the Spanish Association of Lung Cancer Patients, IACAP, and this is also in collaboration with Takeda and Pfizer. So this evening we have a very packed program of really amazing speakers, so hopefully we'll get through everything. And we hope to have time for questions at the end of the meeting, and we'll try our very best to have time available for that. However, we might not get through everything, so you'll have to bear with us. If you would like to type a question, you'll see a little text box, chat box at the bottom of your video screen that you can enter a question into that box. So for those of you who don't know me, my name is Anne-Marie Baird. I am a molecular biologist working in Trinity College in Dublin in Ireland. And of May of last year, I was elected president of LuCE, which is Lung Cancer Europe. And we are a non-profit umbrella organization working at a European level to really be a voice for people impacted by lung cancer and to try to network, advocate, and work any way we can to improve outcomes for people in our community. I also serve as the representative for LuCE on the European Cancer Organization's Patient Advisory Committee. So it's always very important, as much as we can, to give a really strong voice to the lung community in anything that we might do. So tonight, as I said, there's a very packed agenda, and there'll be a number of different topics that we're going to cover, such as clinical trials and treatments, a little bit about lung cancer vaccine, different research lines that are in place, bioinformatics and cancer research, specific needs for those living with ALK-positive disease, and of course then, most importantly, we also have the patient experience as well. So with that, we will kick off with our first talk, which is from Professor Wu. Professor Wu joined the University of California in Irvine School of Medicine out of his Hematology Oncology Fellowship from the Beth Israel Deaconess Medical Center in the Harvard Medical School, and he has now been there for about 18 years. Professor Wu is currently a health science clinical professor at the Chow Family Comprehensive Cancer Center at the University of California, Irvine School of Medicine. He has been involved in the phase 1 development of crizotinib and subsequently early development of next generation ALK TKIs such as alectinib, brigatinib, and lorlatinib. His translational research interests are around overcoming resistance to targeted therapy against both common and uncommon driver mutations in lung cancer. He is also currently an associate editor of the Journal of Thoracic Oncology and the editor-in-chief of Lung Cancer Targets and Treatments. So I'll pass over to you for your talk.
So thank you for the invitation. This is my first time. Thank you. Good evening. It's morning in America, and I'm actually in one of the rooms right here I'm in my clinic, but that's okay. Thank you. I will try to give a little bit of background. The clinical trials, there are many clinical trials. I want to focus on having patients think about actually helping how to design trials in the future, because their input is very important. We do clinical trials, and most of the time the patients are not eligible because of one way or the other. They have too many treatment before, they have these underlying conditions. So I will be very, very quick. And so I like to start out with this diagram. It's an excellent review by Tony Hunter. That was 2001 in Nature. It essentially groups the 58 receptor tyrosine kinases in the human into 20 subfamilies. And as you all know, lung cancer, almost every single one of these receptor tyrosine kinases are altered in lung cancer. You have EGFR, we have VEGF, FGFR fusions, we have targets in bladder cancer and cholangiocarcinoma. TRK, of course, a very hot topic 1 or 2 years ago. MET continues to be very important. And then when you come across to the right, you have RET, you have ROS, you have ALK. So essentially, a lot of this receptor tyrosine kinases will be altered one way or the other in the future. And also, we are finding ligand fusions that binds to EGFR and HER2, especially HER3 and then HER2. You'll hear some of the NRG1 clinical efficacy in ASCO in just very shortly, in about 1 week's time, mostly in lung cancer and also in pancreatic cancer, NRG1 positive. So I want to stress this as we are getting into the almost the second and third decade of ALK positive non-small cell lung cancer. It's very important to identify the variants, because the short variants are much more stable and more resistant, whereas the longer variants less stable in the cell and more sensitive to all TKIs. And that's important now with, yes.
Professor Wu, I'm so very, very sorry to interrupt you, but I was just wondering if you could share your screen so that we can see your presentation, please.
Oh, sorry.
Sincere apologies for interrupting you.
Technology. Let me see. Can you see now?
Yes, that's perfect. Thank you so much, Professor.
Oh, sorry.
No problem. Thanks so much.
So this is the first slide. I will just go quickly. So this is the slide that I talk about all the receptor tyrosine kinases. I won't go back, but here is ALK. You see ROS, you see RET, you see MET, you see EGFR on the other panel. This is the second slide. So this is the variants. So it's important. I know some of the screening, especially with FISH and IHC, do not give you the variants. But as we get more and more into the next generation sequencing, it's very important to know the variants. The short variants are more stable and more resistant, whereas the longer variants are less stable and actually more sensitive to ALK inhibitors. And this is part of the review that we just submitted. So important to know variant 3 and variant 1, they occupy about 35 to 40% of the EML4 positive non-small cell lung cancer. And EML4 positive lung cancer accounts for about 85% of the all positive lung cancer. The others are different fusion partners. And the resistant, I'll show you some very graphic details, very eye-catching details, just like this one. So this is a very nice heat map published by Dr. Christine Lovely at Vanderbilt University. This is part of a paper that was looking at ensartinib ctDNA response. But Dr. Lovely's lab did a very good job. And I'll just look at the top row, variant 1 and variant 3, and just look at the raw number, and then I plot it out on the next graph. So you can see, I'm looking at the 6 all TKIs that are approved in the world. Ensartinib is approved in China as second-line treatment post-crizotinib based on their pivotal study inside China. But you can see variant 3 is in the red bar, variant 1 is in the green bar. So even within each all TKI, there is a difference in terms of the sensitivities. And so that's important, I think, to, especially at the time of initial diagnosis, going forward, knowing the variant status at the get-go, I think it's very important, even for lorlatinib, which, and also ensartinib, brigatinib, alectinib has very good activity against variant 1. But if you look at variant 3, you can see that's a little bit in terms of the reduced sensitivity. And this is all important because 5 years ago, Dr. Yoshida published this paper in JCO, and it led to, I think, subsequent about 10 to 12 papers, retrospective analysis, all showing that numerically, if you have variant 3, the progression-free survival seems to be less. And this is the subsequent another paper, and shows that not just affects crizotinib, but also the next generation ALK inhibitors. And here, and also it's not just EML4. We have in this paper, fortunately, unfortunately, JTO clinical research reports is a new journal. It's not indexed in PubMed, but we look at the published literature as of early 2020. We have about more than 90 fusion partners identified by DNA sequencing. We want to know whether they transcribe into RNA or not, but this is at least 90 I can count on. So it's too big to get the table, but it's important to remember. And here is the structure of the compounds, essentially direct the resistance, because the way they bind, the way it will essentially generate the resistance depending on the binding motif inside the kinase domain. And so this is the timeline. We're very fortunate in the US. We have all the approval, starting with second line, but first line, lorlatinib is approved. So approved last year already. And ensartinib, the data is published, but we haven't heard any approval yet. It's not approved as a second line also. We'll go to Europe. I think lorlatinib has not gained first line approval yet, if I'm not mistaken. It's approved for second line. Brigatinib is approved for first line. So trailing a little bit behind. Japan, they don't have lorlatinib approved for first line. They do have approved for second line. Brigatinib is approved on the same day for both first and subsequent line of therapy. And China, ensartinib is approved. It's the only country that has approved ensartinib based on the Lancet Respiratory Medicine paper published. And so very good data. And I think ensartinib will be approved maybe this year, depending on the exalt-free data and the submission process to the FDA. So because of all that, we have advisory boards and we get passionate debate about which one to start. Should we start alectinib first? Should we start lorlatinib first? We get into a verbal fist fight. Should we get brigatinib? What is the role of ensartinib? So this is how we look at ALK inhibitors right now. It's first, second, third generation. And when you stay at home too long, you get an epiphany, but this is another way of looking at EML4 variants and also looking at the in vitro data of the ALK inhibitors. And here are the bar chart on the phase 3 trials. I'm not advocating one ALK inhibitor over the other. This is just the overall hazard ratio reduction compared to the standard of care, the chemotherapy or crizotinib in the intention to treat population in patients without brain mets and in patients with brain mets. So they all do very well. The hazard ratios are all below 1. So you have a lot of choices, and that's where the debate comes in, and it's very passionate. And so Professor Camich from Liverpool, England, but went to Oxford and Cambridge, wrote an editorial saying that should not be considered. I read the editorial. I was very upset because his attack was very visceral. We did a argument for lorlatinib, but it shows you that there is debate and we don't know, and that affects how we look at treatment in the future. And here is a diagram that when I have lots of free time thinking. So you can do a lot of the scenario. You can start with chemo, crizotinib from the very beginning. You can start with crizotinib, alectinib. You can do brigatinib, lorlatinib. This is some of the thought leaders who advocate. You can jump. You can also do the alectinib, lorlatinib, ceritinib, lorlatinib. Brigatinib, lorlatinib is not quote unquote approved by the FDA. Lorlatinib is only approved for post-alectinib, post-ceritinib. You can also use lorlatinib. And in this case, we don't have any indications, but we predict by resistant that criteria that one of the resistant mutation will be sensitive to alectinib. So there are many ways to do that. And then we put in the diagram here, waiting for ansatinib to get the approval, and we have to remake the PowerPoint slides, but doesn't look like the later it gets approved, the less chance it will be able to get in this crowded field. So not approved. This is the sequence. It's just but in the US, we don't follow the government guidelines that much. And one of the things I want to advocate is if you do sequential treatment, you get into, the first resistant mutation drives the second resistant mutation. This is another of our case report that I managed to do a review in the literature. And these are patient samples we listed on the table. You can see we start out with G1202R. This is the resistant mutation, usually against alectinib, sometimes against brigatinib. And then when you start with lorlatinib, then you have the double mutation, triple mutation, and then we are out of the very important ALK inhibitors. So Jessica Lin from MGH, and we contributed a few patients into looking at combinations of chemo plus TKI. And I always advocate that, even though in EGFR that is not the case where you see additional benefit, but with ALK, you have a very high incidence of CNS metastases, that it is important to, I think, continue. And you can see the one with the dotted line shows this is with chemotherapy. The other one is the one without the dot, the asterisk, are the patients who switch to chemotherapy without continuing the ALK inhibitors. And so these are in the graphic forms. This is a retrospective analysis, but give us at least combinations chemo with TKI I think it's very common. It's just people just don't say it. I just had an email from a very young lady from Malaysia. She's already getting lorlatinib with chemotherapy, and she's asking for help. So that's and I won't. These are the combinations. This is clinical trial. Since the vaccine is going to be mentioned, I will quickly. I want to draw attention to what is happening in the future. The fourth generation ALK inhibitors are coming to clinical trials. What I would call them the double mutant active ALK inhibitors. Not a fancy name. I need to work on that. But TPX0130 and Nuvalent 655 are coming. And here is the abstract from AACR. You can get them from their from their website, download them from the website. One is Turning Point Therapeutics for the TPX0130. The other one is Nuvalent. It's NVL now. It's not N. They changed the name to NVL655. So they can all overcome double mutations and some of them triple mutations. So it's better to download the PDF, and then you can expand and see that. And then how would that affect it? So I think going forward within the next few years, we need to look at reclassify ALK inhibitors as single mutant active ALK inhibitors, and we lump them together. You should go through 2 of them before you get the double mutants, and then you will go into a what we call the double mutant active TKIs. But assuming they are efficacious, they are tolerable, and they have good CNS penetration, which I think they do, based on the preclinical data in the poster. So I'm going to end with the 3 trials because I was thinking about this Labor Day weekend. I didn't go anywhere. It's just too many people traveling. So I want the patients also to think about, how do you develop the next generation 4G ALK TKI? What is the registration trial? You can do a first-line study. I will give you, for most intent and purposes, most people use alectinib. Some people can use lorlatinib. You can put in brigatinib. But at the end of the day, it's probably these 2 the company can, versus the 4th generation inhibitors. This will be a huge undertaking. The PFS from the 2nd and 3rd generation is pretty long. If you want to have the improvement, you will have a lot of trials, a lot of follow-up time, and then you have to supply the comparator arm to the patients to do the trial. I hope they can stratify by the short and the long variants. Detectable ctDNA is important, just like what we do with EGFR mutated lung cancer and TP53, which I don't have the time to mention. Yes, no, it's also important. So this is the first line design. Patients should think about it. Trials should think about it. Sponsors should think about it. This is probably a very expensive trial. You will have to double the amount of patients in the trial, probably about 500 to 600 patients. It will take about 5 years, but this will be the only way you can develop the next generation, because you're not going to go against crizotinib anymore in 2021. Here is a second-line design, a little bit less ambitious. So you do alectinib, ceritinib. I would say alectinib is still it's the most commonly used. Lorlatinib has indication post-alectinib, so this will be the comparison arm. And the 4th generation will be the investigational arm. Lorlatinib PFS about 6 months, 7 months, 5.9 months on the Lancet Oncology paper. So if you want to double or 50% improvement, so you can go from 7 months to about 10.5 months, it's reasonable, it's doable, it's less. And I think this is a reasonable design. So this will be up to the patients and up to the sponsors to see if you agree with this design. Again, the stratification to push us into the next decade, the next 2 decades, on how to find, define, and treat the ALK positive lung cancer. So this is a possible design as a second-line treatment. So that means the first line will always still be alectinib, and these are things to consider. And I will throw in a third-line treatment, since it's specifically designed for double mutations. So you do whatever your first ALK inhibitor, you sequence your second. Everybody can get into including crizotinib, and then you can go at free for all. The sponsor can say just a next generation ALK inhibitors, and then if you have a double mutation, so this one you have to identify double mutation, so you don't have off-target resistance like MET amplification, like other resistance that will not be responsive to another ALK inhibitors. And then you pick your investigational arm, and then here will be chemotherapy, which is considered standard of care. I was thinking on my way there back, INPAL 150, Cabo, paclitaxel, Avastin, Atezo, could be, but this is not the standard at this point. It's a subgroup analysis. It's not approved in the US for this, so it is probably not the standard of care. So this is one way to do it. And then 2-to-1 randomization, because I think continuation of ALK inhibitor is very important to prevent CNS relapse. And then on progression, you allow crossover. So these are things. So I didn't talk about any clinical trials that's ongoing, but the 4G ALK inhibitors are coming. Accelerated approval should be easy or relatively easy relative to a pivotal trial that is required by the FDA in this field and in the EMA and also in other health authorities. So I give you 3 scenarios. You think about it. You can advocate, and then you can see which one is the best, which is the best use of resources and the next development. So I will stop here. My apologies for already over time. Thank you.
No problem. Thank you so much, Professor. That was a really fantastic talk, and it's great to see the things that are coming, both in terms of the new treatments and clinical trials. And I think you made it very clear that there's still so much debate in this area and a lot more that we need to do to find out the best sequencing of treatment and so on for the best outcomes. So thank you very, very much for that. It's my great pleasure to introduce our next speaker, Dr. Carley. So Dr. Carley has to leave shortly after his talk. So unfortunately, he will not be here for the question and answer session at the end. So Dr. Carley, his expertise as a clinical pathologist in areas of hemopathology. Oh my goodness, that's quite the word to get out in the evening. Oncology, immunology, and molecular biology. He received his MD degree from the University of Torino, Italy, and he then went on to complete a visiting fellowship in the Department of Pathology, New York University Medical Center. During this time, he was promoted to a professor of pathology at the University of Torino Medical School in 2014, and he also started a new laboratory in 2012 at the Department of Pathology at the Children's Hospital and Harvard Medical School in Boston. In 2020, he was appointed a professor of pathology at the Harvard Medical School, and he is currently in an attending role at the Boston Children's Hospital currently. He's been leading 2 very busy groups of his own, both in Torino and in Boston, and he has won several awards, and including an award from the Italian National Academy of Science, 2 European Research Council grants, and a huge number of other grants and awards as well. Dr. Carley's major interest has been to study the mechanisms and pathways of tumor formation activated by the ALK oncogene, as well as the development of innovative therapies for ALK-positive cancers, such as ALK-specific cancer immunotherapy. His group has also developed mouse models for both ALK-rearranged lymphoma and lung carcinoma to determine and study the molecular pathogenesis of ALK-driven tumors. Recently, the group have developed an ALK vaccine that instructs the immune system to recognize and eliminate ALK-positive lymphoma and lung cancer cells. And his additional areas of interest are the study of the biological mechanisms of chromosomal translocation formation that initiate those tumor formations. So with that, I'll pass over to you, Dr. Carley, and you are going to talk about lung cancer vaccines. Thank you so much. And please do share your screen.
Sure. Thank you, Anne-Marie, for the very long and detailed presentation. I appreciate. Let me start sharing my screen. Okay, I hope everybody can see that. Okay, and thank you, of course, to the AEA CAP Society, Spanish ALK-positive lung cancer, for this invitation. It's very nice to be able to present our data and the future perspective that we have for the treatment of ALK-positive tumor in general, and of course, in this particular context for ALK-positive lung cancer. After the presentation of Professor Hu, I don't think we need to spend a lot of time to what is ALK, where is found. I just want to share that, of course, in this group, everybody is particularly interested in fusions that drive lung cancer. But as you know, there are many other tumors that are driven also by ALK in the terms of chromosomal translocation, for example, lymphoma, inflammatory myofibroblastic tumors, rarely renal cell carcinoma, breast cancer, colon cancer, and of course other type of tumor by mutation of expression like neuroblastoma, glioblastoma. So the importance of ALK is of course relevant for lung cancer, but is kind of broader and involves many other type of tumors. So any treatment that could help will benefit for patients for bearing this type of alteration in different diseases. And of course, in all these cancer types that are driven by ALK fusion, in particular, this one on the left, the variety of ALK inhibitors that Professor Hu presented are clearly applicable. And actually, they work extremely well. They work very well in lymphoma driven by ALK, diffuse large B-cell lymphoma or anaplastic large B-cell lymphoma. They work very well in stromal tumors driven by ALK. And occasionally, when the fusion is found, they also are also very, very effective in other type of tumors driven by ALK fusion. So the importance of these findings is broader than the lung cancer. But still, lung cancer is by far, by numbers and by frequency, the most important cancer driven by ALK. And why we started to be interested in ALK as a possible target for a cancer vaccine? Well, of course, as a pathologist, we were really driven by several observations, including the fact that the expression of ALK is restricted to tumor cells. And basically, there is no ALK expression in normal cells. And this is extremely important for the immune system, because the immune system and the immune cells likes to see differences. So they want to find something that is expressed by tumor cells, and however, is not expressed by normal cells or minimally expressed by normal cells. And that prevents the possibility of engaging the immune system against normal cells and generating some sort of autoimmunity or some sort of side effects of the vaccine. So in the situation of ALK, we are basically in the perfect situation in which ALK is expressed only by tumor cells and not expressed by normal cells. And that's also testified by the high efficacy of the ALK inhibitor as presented before, and the fact that this ALK inhibitor has all of them very little toxicity, exactly because they, even if they target the endogenous ALK is basically not really expressed very low levels, and so the toxicity are very mild and very well controllable. Another key point for vaccination, and that's we think is a key difference, is that many cancer vaccines have been developed in the past decades, of course, and some of them were somehow successful, at least for a transient period of time, and then they all eventually failed. And one of the most important reasons for this failure was that the target of this vaccine was not an oncogenic driver. And for oncogenic driver, we mean an oncogene that is driving the tumor cells to grow and is present in all of them, basically in 100% of them, which is exactly the case of ALK, because ALK rearrangement is a very early event, is present basically in any single cell of the tumor, and is strongly driving the tumor, right? And this is a key point that because it implies that tumor cells cannot escape the immune system by downregulating this target, because if they do so, then they downregulate the driver of themselves, and so they stop growing, right? So they are basically caught in a situation in which they cannot downregulate ALK, because otherwise they will not survive, the tumor cells. In contrast, in the past, a long list of cancer vaccine kind of failed because they were targeting some protein that simply the tumor cells stopped expressing, and they were proliferating exactly the same. At that point, they became invisible to the immune system. So we think this point number 3 is extremely important for the development of the ALK vaccine. But point number 4 is also very, very important, because another reason for cancer vaccine to fail is that somehow the immune system does not recognize the protein. So not all the protein that we introduce by a vaccine, they generate an immune response. For comparison, in this COVID era, right? At the very beginning, the pharmaceutical company were unsure about the COVID vaccine because they were not sure that the protein that we're using was really eliciting an immune response. Eventually, it turned out to be so, and the vaccine worked very well. So a critical point is to be sure that the protein or the peptide of the protein that you want to use for the vaccine really is recognized by the immune system. It's not silent. So if you give that to the immune system, it will elicit an immune response. And the best way to know that this is hopefully will happen when we will start the clinical trial is that we and others demonstrate that patients that reactivate ALK through a chromosomal translocation, like ALK positive lung cancer patient, they eventually develop spontaneously an immune response to ALK protein. So it means that the tumor cell starts to express the protein, and the protein is seen by the immune system as a new protein, exactly because it's not normally expressed by tumor cells. And this simple expression by the tumor cells is sufficient to activate the immune system and stimulate a little bit of immune response. So the question is, why this spontaneous immune response is not enough? Why immune checkpoint therapy in this patient basically fail? They don't work if this spontaneous immune response is present. The answer that we have is relatively simple. It's basically a quantitative answer. So it means that ALK is immunogenic in this patient. However, the level of immune response that spontaneously the tumor cells can elicit is very low. Its presence is very low. It's low to a point that reactivation by immune checkpoint therapy does not really change the clinical course of the patient. So we think that we need something much stronger, which is a vaccine, to boost this immune response that is happening. However, to bring that to very high level in a way that either a protection or a anti-tumor activity will be finally seen. And that's exactly what we observe in mouse models, and I'm going to present briefly. Okay, so I can skip this because it was already presented before. So these are mouse models that we develop, are transgenic mice that basically we developed by introducing the ML4 ALK fusion, one of the fusion that Professor Hu showed before, one of the most common fusion, the variant 3. So we introduce in the lung of these mice, and you see that very rapidly, in few weeks of age, these mice develop tens of tumors, even hundreds of tumors. We calculated more than 100 different tumors. Yes, this is a slightly different situation than in patient in which you have one single tumor that eventually grows and colonize other sites by metastatic process, but is a good approximation. And what we observed that given a simple vaccination was very, very rapid vaccination, too short of vaccination, this vaccine alone was able to contain the growth of the same tumor. So this is a tumor that is already present, and then you see that does not grow over time. In contrast, for example, for this tumor in non-vaccinated mice that grows and becomes bigger and bigger and eventually kills the mouse. But not only that, basically the vaccine alone was able to prevent the development of so many tumors in the mouse. And below is a quantification. And so we show that vaccination alone was able to do a strong job. And we even measured the number of immune reaction inside tumors. And probably this slide is simple enough to be understood rapidly. So on the left is a tumor without a vaccine. And you see that these are the tumor in mice. And there are very few, these brown cells are T cells in the tumor, very few of them. However, after vaccination, look how many T cells from the immune system go inside the tumor. And of course, the tumor is much smaller, shrink down because it's killed by the T cells that infiltrate the tumor. And we calculate this extensively. However, as just presented, the classical therapy for ALK-positive tumors, of course, is treatment with ALK inhibitor. We use crizotinib at the beginning. Now we are using lorlatinib. We wish we could test all the ALK inhibitor in mouse model. It will be a huge number of experiments and expensive, so we need to select a couple of ALK inhibitors. So we did both crizotinib and alectinib, first and third generation. And you see that in mice, crizotinib works exactly like in humans. So you have a tumor there. The tumor basically disappears. However, is not cured because eventually the tumor comes back exactly in the same position and grows. And then there are secondary tumor also upcoming. But look at the vaccine. So the vaccine, for example, you start with two tumors like this. You treat with a combination of crizotinib plus the vaccination. These two tumors go away and they never come back eventually. Very small tumors later on, after a few months, they grow, but this is the technical situation of the transgenic mice, because the transgenic mice, every single cell in the lung has the ALK fusion. So potentially every single cells in the lung, which are millions and millions of cells, can become a tumor. And after so many months, eventually the immune system also becomes exhausted. And so we did not vaccinate it again, but we can vaccinate again and revitalize the immune system to provide more immunity over time. Okay, and here basically, these are the conclusions that we learn. I don't want to read them because I think I gave enough information. So how we want to move this finding in mice toward a clinical vaccine, and what are the steps that we are doing? So first of all, in collaboration with Dr. Mark Awad at the Dana-Farber Cancer Institute and also Dr. Pasi Janne, still at the Dana-Farber Cancer Institute, we demonstrated that, as I said, spontaneously, these are in blue, these are all lung cancer patients in which we detected a spontaneous ALK immune response. So you see that there is about 20% of them that develop a quite strong immune response, but most of them, they develop a very weak, almost undetectable immune response. So this is where the vaccine, the plan is to boost this immune response to level that will be therapeutically efficient. So how to move to the clinical world? So of course, we need to have evidence that a vaccine formulation based on peptides, which is the typical way to vaccinate patients. And I can immediately answer your question, why don't use other type of vaccination? We learned from COVID that peptide vaccine is a possibility. There are also viral vaccination. There are also mRNA type of vaccine. Yes, it is possible. However, for example, mRNA vaccine that work very well for COVID, they still don't work so well in tumors. And remember that both companies that develop COVID vaccine, Moderna and Pfizer BioNTech, they were eventually company built to develop vaccine against cancer. Then they switched to COVID because of the situation and the emergency and the opportunity, of course. But both companies were founded to find cancer vaccines, okay? So mRNA vaccine is a possibility. We have not tested that. Maybe in the future will. However, it is likely that works very well with big proteins, not with small peptides. So the classical way to vaccinate now in 2021 is to use long peptide. Long peptide means a small portion of protein that are about 20-25 amino acid long. So to develop a preclinical concept that is very helpful to file at the FDA level an approval for this clinical vaccine and start the trial. So we identified the immunogenic ALK peptide in the mouse model, and then we basically tried a clinical trial in the mouse. And the clinical trial in the mouse is vaccinating mice together with an ALK inhibitor, lorlatinib in this case, because I will show how the clinical trial design will be. And we can add immune checkpoint therapy to potentiate eventually the vaccine response. And these are the results. You see in black is mice that are untreated, so they die very rapidly of this ALK-positive tumor. When you use lorlatinib or lorlatinib plus immune checkpoint therapy like PD-1, you extend survival for quite a bit. I mean, you double survival. You see that it's almost double, goes up to 50 days. However, all the mice invariably die. So lorlatinib alone and lorlatinib plus immunotherapy is not sufficient to cure mice or to extend significantly survival. However, when you start to combine the vaccination, we can go from the best, which is this gray line. You see that we can basically completely cure forever about 80% of mice when we combine lorlatinib plus vaccination plus this immune checkpoint therapy. But even lorlatinib plus vaccination alone, forget the immune checkpoint therapy for now, which is the red line, you see that we shift even more the survival curve, and we are able to cure about 30 to 40% of the mice, which is a big achievement if you consider that no mouse is cured by lorlatinib or lorlatinib plus immune checkpoint therapy alone. So this data will be fundamental for the application to the FDA. So how we want to translate this one to the clinical, in collaboration again with groups at the Dana-Farber Cancer Institute, Kathy Hu, Ellis Reynolds, which work on mass spectrometry and peptide identification, Mark Award, who provided patient samples. We've basically, to make the long story short, it took several years to get this data. We were able, by mass spectrometry, which is a technique that identifies peptide presented by the tumor cells, we were able to identify 4 ALK peptides that are presented on the surface of the tumor cells. So they are there. They are on the top of the cells, and they are recognized by the immune system. And in fact, we proved that using this peptide, we were able to vaccinate humanized mice. In this case, everything is human. So it's the human immune system and the human peptide. So we were able to vaccinate them. And on top of them, we were also able to show that, look at, for example, this patient number 4. This is a patient from a series of patient treated at the Dana-Farber. We took the peripheral blood and we incubate the peripheral blood T cells, those the lymphocytes of the immune system, with this peptide that we identify. And look how good is the response. This is without vaccination. This is the spontaneous immune response. So it's very strong, even without any vaccination. So we hope that by vaccinating, we can make this response 10 times stronger and finally get to a clinical activity. So the clinical trial design, and this is basically the last slide, the clinical trial design will be 2 cohorts. The difference is the cohort number 1 is the vaccine alone. Cohort number 2 is the vaccine plus immune checkpoint therapy. And we will, each of these arrow is a vaccination. So will be many, many vaccinations to potently boost the immune system, and will be conducted in collaboration with this company, Elysium Therapeutics, which is supporting the vaccine manufacturing. They have also a program in KRAS. Now they also a program in ALK. We are still in the preclinical phase, and we hope that by next year we will be able to open and get the FDA approval to open the clinical trial. And this is the group I already thanked most of the people, in particular the Dana-Farber group, Mark Award, which is our strongest collaborator. Okay, I think I can finish.
Thank you so much. Thank you so very much, Dr. Carly. That was a great presentation and very, very exciting to see that preclinical data, and fingers and toes crossed for the upcoming clinical trials, because I think hopefully it will make a big difference. So I'm very conscious that we are running over time a little bit already. So without further ado, I'll introduce our next speaker, which is Dr. Martini. He has over 20 years of experience in the biotech and pharma industry in pharmacology and clinical translational scientists, and is the author or co-author of over 50 scientific publications. Dr. Martini is currently the Senior Director of Translational Oncology and Global Product Development and Oncology, and has been at Pfizer since 2011. He currently leads or supports translational strategies for Ibrance, lorlatinib, my goodness, the words deceiving just don't want to come out. He also leads the cross-functional efforts in the area of liquid biopsy, circulating free DNA, where he represents Pfizer as part of the Blood Profiling Atlas in Cancer and the Friends of Cancer Research. He also heads the translational strategy for Pfizer-sponsored Translate study and is the Pfizer Board of Director representative for the WIND Consortium. So it's really lovely to have you here this evening, and I'll pass over to you for your talk, Dr. Martini.
Thank you, Anne-Marie, and thank you. I thank the organizers for inviting me to participate to this webinar. Really appreciate the opportunity.
I'll just get you to share your screen, if that would be okay, so we can see your presentation. Thanks so much.
Is this showing properly? This correct. So today I will discuss, these are my disclosures. I am, of course, an employee of Pfizer and I own stocks in the company. I will be discussing a drug that actually Dr. Wu and Dr. Cherry already mentioned quite a few times, lorlatinib. Lorlatinib has received a conditional marketing authorization in Europe. And today I'm going to present some data from a clinical trial. The questions I will be addressing will be the following: some input on liquid biopsy, which is a key topic of mine. The implication for ALK positive lung cancer. We will go through some history of Pfizer implication in this specific disease. We'll set up the question and we'll go over some data that we presented a couple of years ago by Dr. Shaw at ASCO, and also move on into some perspective and set up for actually this year's ASCO, where we'll be presenting additional data. So liquid biopsy or cell-free DNA is definitely making a significant advancements in the past few years. And as you know, circulating DNA is coming from multiple areas of the body. Your normal cells are actually shedding some of that DNA when they die, but tumors actually, due to the turnover of the cells and these cells also dying, tumors are releasing a DNA in the bloodstream, and that DNA can be later harvested and then analyzed or sequenced to understand what are the molecular events that are represented in the tumors. The advantage of those liquid biopsies, of course, are that they are minimally invasive. There is less risk associated with the collection of these specimens. Venipuncture is relatively low cost compared to an actual true tumor tissue biopsy. Safety, again, is definitely possible within pretty much all patients. In addition, there is the possibility to look or collect specimen upon treatment. And it's more difficult to have serial tumor tissue biopsy, as everyone knows, and more cumbersome for the patient. So definitely not something that's done on a regular practice, while a blood draw can be done on a more regular practice. Disadvantage, because there are some, it's less sensitive than doing the analysis on tumor tissue. There is a greater risk of false negative, and currently we are limited at the analysis of the DNA, but there are some other new platforms that are coming up, looking also at RNA coming from the tumors, and also looking at other aspects of DNA, not only the mutations per se, but also methylation on the tumors, on the tumor DNA. So in terms of the history, Pfizer has been very much involved in ALK positive lung cancer for the past decade. And actually, with the initial trial, the Profile 101 trial of crizotinib that started in 2006, 15 years ago, actually this year was the anniversary and the study closure of this Profile 101 trial. Starting in 2006, crizotinib started in the clinic as a MET inhibitor. And then by 2007, publication in Nature by Professor Mano from Japan described this EML4 ALK fusion. And that was the seminal work that triggered the whole change in perspective from the ALK positive lung cancer. Based on this publication, Pfizer updated the protocol for this Profile 101 trial. The first ALK positive patient was enrolled in the fall of 2007. The first response was observed for such a patient in early 2008. And then the rest is history. In 2011, there was the approval of crizotinib for ALK positive lung cancer. This approval happened in May of 2011 in the US. It was followed over a year later with the approval in Europe by the EMA. And full approval came even further later in 2015. And then also approved for ROS1 eventually in 2016. And as you can see on the slide, there are multiple diagnostics that have come up along the way, such as the initial Abbott-Fish test that Joe mentioned, as well as some PCR assays, and then the IHC assay from Ventana, and then some new next generation sequencing platform that came up in the past 5 years. Along these, Pfizer continued its work for this specific group of patients, knowing that there were mechanisms of resistance that were identified early on, such as an increase in the rate of brain metastasis in patients treated with crizotinib. In addition, there was the emergence of ALK kinase domain mutation, such as the 1196M mutation, that was also observed early on in patients treated with crizotinib. And Pfizer worked on establishing a new drug that would be able to address both issues, the resistance mutation and the brain penetration. Came along lorlatinib, which was first introduced in the clinic at the end of 2013. In the end of 2016, early 2017, lorlatinib gained breakthrough therapy designation by the FDA. And in September of 2018, based on the result of the original trial, lorlatinib was first approved in Japan and then approved in November of 2018 by the FDA in the US, and then further in May 2019 in Europe by the EMA. Lorlatinib was also looked at in first-line treatment in this phase 3 trial that Dr. Wu mentioned. And based on the result of the study and the interim analysis that occurred in the summer of 2020, lorlatinib gained an extended approval and a full approval by the FDA in the beginning of March of this year, and we are awaiting decisions from the EMA for Europe. So the question that I'd like to address today is, can early monitoring of circulating DNA or via liquid biopsy for lung cancer help monitor treatment response to patients with lung cancer? And what can we learn from the ALK positive lung cancer cases? We know that there is a lot of things going on in the realm of liquid tumors. So this is definitely the case of this minimal residual disease assay that has been approved by the FDA for ALL as well as multiple myeloma. There are a lot of efforts in the community through various consortia to try to establish for solid tumor whether or not this concept of minimal residual disease can be utilized to monitor treatment response by patients. And this is what we're going to be discussing next. So this was presented at ASCO in 2019 by Dr. Shaw, as I mentioned. This was coming from an international study with a representation from France with Dr. Bess, representation from Spain from Dr. Philippe, and representation from Italy with the Dr. Chiari. And of course, actually, Dr. Wu was also one of the investigators on the study. This was the different cohort that were looked at in this study. I will focus only on the ones that are depicted with the red square. Those are patients who have received multiple lines of therapy, either a second generation ALK inhibitor, such as alectinib or ceritinib, or patients who had received 2 prior ALK inhibitors, such as crizotinib and a second generation ALK inhibitor, or patients who had received 3 prior ALK inhibitors, such as a sequence of crizotinib, ceritinib, and alectinib. What we looked at is actually circulating DNA. So looking at plasma collected at baseline, as well as about 6 weeks on treatment, we harvested the plasma and analyzed the circulating DNA utilizing this Guardant Health G360 platform. And we looked at the mutations and other aberrations that are carried on the tumor DNA to ascertain variant allele frequency, so the amount of those aberrations, either strictly related to ALK, so the ALK fusion and the ALK mutation, or aberrations that are encompassing any type of genes, not only ALK, but as Dr. Wu mentioned earlier, also TP53 and other genes. And we looked at various ways. First, the average of those variants frequency at baseline and at week 6, and also the difference between what's observed at week 6 versus what was present at baseline. And we correlated this with the response and PFS and OS. This is a consort diagram of the samples that were collected. I'll go very quickly through that just to say that, as I mentioned, there are definitely caveats in utilizing these liquid biopsies, because not all tumors are shedding circulating DNA. And as you can see here, while we had 121 paired sample between baseline and cycle 3 day 1, only 88 of those paired sample had actually any type of alterations. 33 or slightly over 25% did not have any aberrations to follow. So we could not do much with these, but these have very specific biology. And it is also important to have this information. It looks like these tumors are behaving differently than the tumors that are shedding circulating DNA. And within those who are shedding circulating DNA, we had 57 that had ALK events and 31 that did not have any ALK events. So in this specific cohort, most patients were heavily pretreated, as I mentioned, and had received at least one prior second-generation ALK inhibitor, such as alectinib, ceritinib, or brigatinib. Circulating DNA was detected in 73% of the patients with paired sample, and 53% of the patients had an ALK event that was detectable, or ALK fusion that was detectable at baseline. Baseline characteristics for all those patients, for patients who had different function in terms of modulation of their circulating DNA, were similar, including in terms of location of the involved site of disease, as well as a number of sites of disease. So it's very important. We are not looking at different populations here. It's a very homogeneous patient population. This is a diagram that depicts how circulating DNA evolved during the course of a treatment between baseline or screening and week 6 or cycle 3 day 1. And you can see that the majority of patients have a drop in their circulating DNA content, but those patients have either only stable disease or progressive disease have actually an increase in their circulating DNA. This is more or less expected, but that means that there is a correlation between what is happening with the size of the tumor and what is happening with the amount of circulating DNA that can be detected in the bloodstream. And this is further represented in this diagram, where you can see that patients who have a decrease in the amount of circulating DNA that is measured at week 6 versus baseline have a better decrease in the tumor volume as assessed by RECIST or by radiographic measurements. You can see that these patients in general have a reduction of their tumor measurements by about 26% from baseline, while the patients who have an increase in the circulating DNA amount have only a slight decrease in their tumor volume of only 12%. And this is more going into the weeds, but basically what we are trying to demonstrate here is that for patients who have a strong decrease in the amount of circulating DNA detected at week 6 versus baseline, there is a strong correlation with the response status. And actually patients who have progressive disease or no response at all have actually an increase in the amount of the detected circulating DNA. And this is particularly true when we look specifically at ALK fusion or ALK mutation. When we look at any type of alteration in any genes, you can see that the correlation still exists, but it's a little weaker. Now, looking at progression-free survival, there as well, there is a good correlation between the amount of decrease of circulating DNA that's measured at week 6 versus screening, patients who have a strong decrease in that amount of circulating DNA, it's the white line, are doing better than the patients who have an increase or no change in the amount of circulating DNA detected at week 6. And it's actually significant. You can see that the median PFS for patients who have a decrease is about 6.6 months versus the patients who have an increase or no changes. The median PFS is only 2.6 months. This actually also is reflected in terms of overall survival. For patients who have a strong decrease in the amount of circulating DNA, the median overall survival is 18 months, while for patients who have an increase or no changes, the median overall survival is only 8.6 months. And now if we look a little further, if there is actually clearance, meaning that at baseline, you can detect circulating DNA, especially ALK fusion or mutation, but then at week 6, in the patient's blood sample, you cannot anymore detect this circulating DNA, which is this mean VAF of 0, or the second bullet from the top. The median PFS becomes 11 months, while those patients who have still some circulating DNA detected, actually the median PFS is only 4 months. So there is definitely some very good biological correlation here between the decrease in amount of circulating DNA and the extent of the benefit derived by the patients. This is very true when we look specifically at this ALK fusion and mutation. It's less true when we look at all the combined molecular aberrations that are detected in the circulating DNA, which is depicted on the right of the slide. So in terms of conclusions, early circulating DNA dynamics may predict lorlatinib efficacy in ALK-positive metastatic lung cancer, and decrease of circulating DNA at week 6 is associated with better response, longer PFS, and longer OS. This is particularly predictive when we look specifically at ALK fusion or mutations. It is less so when we look at the overall molecular aberrations that are detected in the circulating DNA. Further studies are needed to validate those findings and determine whether early intervention based on circulating DNA monitoring may improve outcome. It's based on this work that actually tomorrow, Dr. Su from Singapore will be presenting data from the Crown study, or phase 3 data of lorlatinib at ASCO. It will have an oral presentation showing even further analysis, deeper analysis of this concept of minimal residual disease, as well as this concept of molecular response based on circulating DNA. In terms of conclusion and perspective, collection of the right biospecimen at the right time is critical. Tumor biopsy collection is a challenge. It's still required because we definitely do learn more from the tumor tissue than we do from the circulating DNA. However, liquid biopsies are more feasible, easier to do, and it can help monitor disease evolution as well as treatment effects. The appropriate clinical application of circulating DNA or liquid biopsy technologies continues to evolve, and patients should definitely be front and center of our efforts, and more collaborative approaches are needed. And we've seen that actually even during the pandemic and the curfews, there were still possibility of working with the patients, for the patients, to get those clinical trials to completion and get submissions for the next lines of therapies. With that, I would like to thank the study participants, their family, their caregivers, all the investigators, and some of them I have mentioned, their co-investigators, the study team, and of course, my colleagues at Pfizer. Thank you.
Thank you so very much, Dr. Martini, for that great presentation. And you're absolutely right. It's so important that we continue to collaborate and work together for the benefit of the community. And it's great to see more and more research being undertaken in the liquid biopsy space. So thank you very, very much for that. So we'll move on now to our next talk, which is on bioinformatics and cancer research. And it's my pleasure to introduce Dr. Brooks. He's graduated as a pathologist in Antwerp University in 2019, and since then he's been a member of several national and international working groups. His main interests are in molecular pathology, digital pathology, artificial intelligence, and breast and gynecological pathology. Dr. Brooks has participated in several projects concerning computer modeling for therapy prediction and artificial intelligence related to cancer cases to seek the full implementation of bioinformatics in pathology practice. So thank you very much, Dr. Brooks, and I'll pass it over to you for your presentation.
Thank you. Thank you so much for this kind introduction. I'm going to share my screen now.
Thank you.
So many thanks for the introduction again, and also many thanks to all the organizers for inviting me to speak at this online seminar. For the next 50 minutes, I would like to talk about bioinformatics and how it helps us in cancer research, but also how it helps us in clinical practice. And bioinformatics is something I'm very passionate about, so I'm very grateful that I may talk about this. Unfortunately, I only have 15 minutes, but bioinformatics is something I can talk about for hours. So first, what is bioinformatics? Bioinformatics is simply the application of computer science to biological data, for example, next generation sequencing data. And bear in mind that it's actually not the same like bioengineering. Bioengineering is actually the science of building biological computers. The main goals of bioinformatics are to manipulate and visualize raw biological data, such as next-generation sequencing reads, and to transform this into interpretable output. Furthermore, this interpretable output can also be used to perform biostatistics on. And finally, bioinformatics is also involved in the storage of this data in databases and data frames. And if we talk about bioinformatics, you will always hear two terms, one being processes and one pipelines. But processes are what their name indicates. They process input to output, and a pipeline is just a sequence of different processes where the output of a leading sequence is used as the input for the following process. And for that, I'm going to get my pointer. I have an example here in the right bottom of my slide. This is just a Linux command. And in the last example, we just add another process to a pipeline. All the commands are separated by a pipe sign. And this last addition just asks us to use the output of the previous command, which is shown here at the top, to just print the 9th column, and you get this. Bioinformatics is a very broad computer science and comprises the knowledge of operating systems, databases, especially public biological databases, a lot of programming languages. But luckily for us, for some recurrent problems, specific tools already exist, as you can see at the right bottom of my slide. And they exist in the form of software applications that are ready to use for the researcher. But what is now the exact role of bioinformatics in cancer research? Well, there are many applications for bioinformatics in cancer research, but as I will only talk about this topic for 50 minutes, I will limit myself to specific applications. And the first application is actually not only found in cancer research, but already in clinical practice as well. Because even in clinical practice, we already try to determine the mutational status of genes and proteins. But how exactly do we go from the resection, as we see on the left side, of a lung tumor, to the knowledge of, for example, a mutation in EGFR or a fusion in ALK? Well, the answer to this question consists actually of two parts. On one hand, we have our biotechnology, such as next-generation sequencing or fluorescence in situ hybridization. But on the other hand, we also have bioinformatics tools that helps us to interpret all those biotechnology data. And for the next couple of slides, I would like to demonstrate this on the example of next-generation sequencing. Next-generation sequencing, as you all probably already know, is when we have isolated tumor DNA, we link this with adapters in the library preparation phase. And an important purpose of these adapters is actually that we can identify this in the bioinformatics phase. Next, the strands are hybridized and amplified with the sole purpose to have an optimal signal in the final phase. In the final phase, the real sequencing starts where the single strands are bound to fluorescence-labeled nucleotides, as you can see here in the top. And for each cycle, the released fluorescence signal when a nucleotide is bound is captured. And from here on is actually where the bioinformatics plays a role. At the end of the sequencing, we only have actually raw biological data, which only consists of the color signals. And for that, for the interpretation of these signals, we still need the whole bioinformatics pipeline to get an interpretable output, such as a report. And for this, we have a whole bioinformatics pipeline that you can see here on this slide. But for the next couple of slides, I will try to summarize this in a more simple form. So where do we all start? At first, we have the signals of our next generation sequencing reads. And these color signals are captured for each cycle. And each cycle is just a next nucleotide. But what we also capture is the quality of the signal. And all this data is getting stored in a FASTQ file, which consists of the identifier, the sequence, and our data of our quality scores. Well, we end up with a text file. And the beauty of a text file is that we can easily process this file to search for things that, for example, a DNA mutation. And that's what is done next. So next, the sequence that is available in the FASTQ file is aligned to a reference genome, and it is stored in a SAM-bound file. And finally, the SAM-bound files are used to search for variations against the reference genome, and this is stored in a variant call format file, which will store the presence of the DNA mutations. And finally, these files can be used to visualize or visually align the reads to a reference genome in order to have a good overview of the read depth and the coverage of the variants and the genes. And in the end, the only thing that we still need to do is to verify if the found variant is pathogenic or not. And that we can do by comparing this to databases that are available. And this is, for example, an example of the COSMIC database in which the BRAF V600E mutation is shown. And after this, the only thing that is need to be done is to report the variant as a matter of data visualization. So the previous example is something that we do already every day in clinical practice. So it's not just research anymore. And I know that the title of my presentation is Bioinformatics in Cancer Research. But bioinformatics also enables us to take cancer research to a higher level. And assuming that we already have our knowledge on the mutational status of a protein, the next step that we can do is to create three-dimensional computer models of this mutated protein to investigate the interaction properties between different atoms. But on the next level, we can also add a compound such as a ligand or a TKI molecule. In this example, we added crizotinib to a ROS1 molecule, and we try to explain in this in silico models how well the compound interacts with the 3D molecule of the protein that has been mutated. In literature, you will find several terms referring to this research area, such as molecular docking, molecular modeling, and molecular dynamic simulations. Well, they are mostly synonymous to one another, but where molecular docking is actually the field where we try to explain where the ligand can bind, molecular dynamic simulations will rather tell you how well this binding is. And in our own research department, I work with Farhan Ulhak and Geert van de Weijer, who are the principal investigators of this research that is performed. I know this is done on ROS1, but it can be easily translated to ALK as well, and is a possible future perspective of our research department. So what they basically did is to determine the RMSD between of all the atoms. What is RMSD? Well, it's actually the average distance between the same atoms of the same positions between before and after the introduction of the compound, in this example, crizotinib. So it actually just tells us how the molecule, how the atoms are displaced in space. And we determined this for the addition of crizotinib for several mutated ROS1 receptors. Well, several ROS1 molecules, such as the ones with mutations at residue 9051, as you can see in the third one, and 2101, as you can see in the fourth one, shows a significantly difference in RMSD, as you can see here by the red line in comparison to the black line, which is the wild type ROS1 molecule. And furthermore, this research has shown by comparing the RMSD for each residue of the wild type ROS1 molecule and the ligand-bound ROS1 molecule, we were able to determine the most important residues, as you can see here in this plot, that vouch for the successful ligand binding, such as crizotinib, as illustrated in this figure, where you can see crizotinib with in the interaction with the most important residues. And I know this research was done on ROS1, and I also need to talk about the ALK positive disease, but I can show you this one of many articles that are already available, and I just took one of the first ones. In this one, they have been doing a similar research for the ALK molecule, and this article demonstrates molecular modeling of ALK in combination of several tyrosine kinase inhibitors, as you can see in the right bottom of my slide. And based on this research, the author was able to determine the most important residues for ligand binding, for example, for crizotinib, as you can see here, as you have been seeing on the previous slides for ROS1 in our own research. But what they also determined is the most important residues that are important in case of resistance mechanisms for several different tyrosine kinase inhibitors. Well, this research field is still highly investigated. It's a really hot topic at the moment. Well, as a future perspective of this research field, I anticipate that the use of these models can aid in design of new inhibitors, in that they can help in overcoming current and possibly in future molecular resistance mechanisms that each molecule can induce. And then another important aspect of bioinformatics in research is the possibility to use powerful tools in context of biostatistics. For this task, we have our project. It's a very easy to learn programming language. And well, it is used for data analysis, data visualization, but also you can use packages to perform very powerful biostatistics as well. The importance of R in literature can be seen by a publication that has been published by Hickman in Nature, where he tried to search for how many publications mentioned the use of R in their research. And you can see that almost actually for all of the research areas, there is a steep increase in the use of R in the publications. And finally, I would like to quickly mention another important research area in bioinformatics. Alas, I don't have a lot of time left to talk about it in detail, but I cannot forget to talk about it as well. So artificial intelligence, it is an area that is involved in investigating computer processes to do tasks that normally require human intelligence, such as visual perception, decision-making, and communication. And well, there are two main types of artificial intelligence. We talk about supervised machine learning when humans are still needed for feature extraction, on which machine learning bases decisions. But on the other hand, we have also the unsupervised learning, where the computer will extract all features itself. Well, explaining artificial intelligence will require several hours, so I will not go further into detail on this, but the message that I would like to give is that artificial intelligence helps us processing large and complex data, which can help us also in the previous applications that I've shown you earlier in my presentation. And this brings me to the end of my presentation. Well, in summary, bioinformatics is becoming more and more important in the research and also in clinical practice, because we use this already for the interpretation of next generation sequencing data. At this moment, molecular modeling is a very hot topic in bioinformatics cancer research, and it is anticipated that this can be used in the help for designing new drugs that can overcome molecular resistant mechanisms in ALK ROS1, but also in EGFR, HER2, and other molecules across different cancer types. Finally, bioinformatics is also used for biostatistics, and artificial intelligence is also a very important part of bioinformatics. So in the end, the importance of bioinformatics to me is that we can integrate this tool in, or these tools in clinics to help therapy decision-making and to improve the patient's prognosis. And that's what it's all about in the end. And I would like to thank you so much for your attention.
Thank you so very much, Dr. Brooks, for that. And absolutely, completely agree. There really is a need for better integration of bioinformatics. Particularly, I think we need a lot more of clinical bioinformaticians as well to help look at that NGS data. And as you say, in the digital pathology space, I think we're really only at the beginning there to see what the impact of AI can be on that. So very exciting area, and thank you so much for that overview. So our next speaker is Dr. Bonatti, who's going to talk about specific needs of people living with ALK-positive disease. Dr. Bonatti is a consultant medical oncologist at the Santa Maria della Croce, I hope I pronounced that correctly, hospital in Italy. Dr. Bonatti is a specialist in lung cancer with a focus on targeted therapies in immunotherapy, and her research interests include promoting physical activity to prevent the risk of cancer and to improve the quality of life of people who are on active treatment. Dr. Bonatti, I'll pass over to you, and I'd just like to remind you to share your screen, please. Thank you so much.
Yes, thank you. Good afternoon to everyone. While I'm trying to share my slides, I would like to thank all the organizer and the association, the Spanish Association of Lung Cancer Patients for this. I don't know if you can see my slides.
No, not yet.
Okay, I will try again. So let's see if I'm able to show them. Okay.
Maybe if tech support were on the line, if they could maybe assist Dr. Bonatti.
Let's try again. Okay. I cannot see it. I'm trying to share, but I cannot see my presentation. Let's try again.
So just from the share screen at the bottom, it's not sharing, is it not?
Yeah, I can see it. I can see the screen, but I cannot. Okay.
Perfect.
Yes,
we can see them now.
For the delay.
No problem at all. Thanks so much.
Let's see. So I would like to thank the Spanish Association of Cancer Patients and Lung Cancer Europe for this unique opportunity to discuss about special needs of lung cancer patients with high rearrangement. And I would like to deepen onto a topic which is not often addressed in oncology, which is physical activity in lung cancer. Why do we need to talk about physical activity in cancer? Well, because muscle derangement and malnutrition often occurs in more than one half of patients at disease presentation. And malnutrition is related to a combination of factors such as for reduced food intake and muscle and metabolic derangement, either host or tumor related. And they're often reported during chemotherapy and radiotherapy, and also with tyrosine kinase inhibitors, which are the drug of choice for high positive non-small cell lung cancer patients. So why do we need to prescribe as oncologists the physical activity? Because physical activity and exercise represent a non-pharmacological intervention that have been shown to have been proven to reduce fatigue, cancer-related fatigue, which is also a frequent adverse event related to tyrosine kinase inhibitors, to improve quality of life, to improve muscle strength and muscle mass, and to reduce anxiety and depression, which are related to a diagnosis of cancer. And of course, general advices cannot be applied to all patients, to the whole population of non-small cell lung cancer patients, and specific guidelines do not exist yet. So our group is working a lot on promoting physical activity, first of all on prevention. There are lots of data in literature, and this is one of the largest one is a pool analysis, including more than 1,000 patients that show the high level versus low level of physical activity may reduce the risk of 13 kinds of cancer, and higher level may be associated with a reduction of more than 20% of risk of 7 cancer, which are the ones with the asterisks. This is important because this led the oncologists to think a lot about that, because, you know, when we offer chemotherapy to a patient which has been submitted to a surgery, we know that the survival advantage is really poor, is about 5%. So this doesn't mean that we should not offer adjuvant chemotherapy, but probably we should add in our prescription, together with chemotherapy, also physical activity, physical exercise. This study is important also because it showed that the greater advantage was for those people who have normal weight and are non-smokers in smoking associated tumors, because physical activity, of course, is a part of a correct lifestyle, which includes adequate nutrition and smoking cessation. But a small benefit there was also for people who is overweight and with obesity, and these findings may help engage people overweight to be physical active, and this is important. So what is the role of physical activity when we have a diagnosis of cancer, after a cancer diagnosis? Well, we have data about breast cancer, prostate cancer, and colon cancer that show that physical activity may reduce the risk of recurrence or death for any causes, or of a range from 30 to 60%. How much physical activity we should be doing? Well, it's not a big commitment, just a small commitment can be enough. As you can see, working in the study of breast cancer, the Narcissus Health study, working from 3 to 2 hours a week at an average pace, that means that we can run a conversation with a friend and we're working with, may reduce the risk of recurrence of breast cancer by 40–50%. So just a small commitment is enough. Another important point is that this advantage in reducing risk of cancer, risk of relapse of disease, and risk of death was present also for people who have never been physically active in their life. So another important point that is never too late to start physical activity. Also, if an individual have never been physically active before the diagnosis. Listed here are the goals of exercise for cancer survival, and I will focus my attention in fatigue because, as I said before, this is one of the most frequent adverse events, cancer-related, but also related to treatment that we gave to our patient, included the tyrosine kinase inhibitors, the one that the professor who listed before, so crizotinib, sartinib, alectinib, lorlatinib. The most common adverse event is fatigue. Fatigue may be exacerbated by other symptoms like neuropathy, like renal syndrome-like symptoms, anxiety and depression, and may last for years also for people who have been cured for cancer. It lasts, for example, in 33% of people who have been cured for breast cancer. So no drugs are available for fatigue except for physical activity and psychological support. Of course, physical activity should start slowly and gradually enhanced in our patient. In the era of tailored therapy, in the era of personalized therapy, also physical activity should be personalized in a multimodal treatment, in a multimodal management of our patient. In physical active, just from 3 to 5 times a week doesn't mean that the rest of the time we can stay on the couch sitting. Of course, because sedentarity is an independent risk factor for cardiovascular disease, such as stroke, but also for cancer and diabetes. So if we know that we're going to spend a lot of time at our desk in front of the computer, because we know lots of patients with hyperarrangement are able to work because they have a tremendous quality of life, we should take some tips to adopt. For example, if I know that I will stay at the desk for several hours, I should get up every half hours and take some steps, prefer the stairs instead of the elevator, go bicycling to work. There are some tips that can help our lifestyle. These are the American College of Sport Medical guidelines for exercise and cancer. And to summarize, we can say that 90 minutes per week, which means 30 minutes to 3 times a week of aerobic activity in combination with resistance exercise 2 times a week may reach all these outcomes: cancer-related fatigue, quality of life, physical function, anxiety, depression, and lymphedema, which is one of the most adverse events after an axillary lymphadenectomy in breast cancer. So again, a small commitment is not too demanding, is never too late to start. Just take in mind this kind of points. Our efforts in promoting physical activity in cancer patients who are in active treatment resulted in this study that is going to enroll patients from July 2021. The excellent study is a phase 2 feasibility randomized study, which will enroll patients with ALK translocation, BRAF mutation, EGFR mutation, and RET translocation, people who have a metastatic disease, and will be randomly assigned to ARMA, which is a supervised home-based specific program training of exercise or general counseling. Primary endpoint of the study will be quality of life, and amongst secondary endpoints, we will have impact of physical activity, intolerance to cancer therapy, because you know that physical activity may enhance cancer therapy tolerance. And of course, an exploratory one, patient will be submitted to blood sampling once a month to verify the modulation of immune system during physical activity, because we have lots of data that demonstrate that with physical activity we can have an enhancement and an activation of T lymphocytes, which are one of the most important barriers of the immune system against cancer. Of course, I put this slide because I should always remind to myself that I should be the first good example for my patient. If an oncologist is physically active, he has more chance to have physically active patient, because he is able to counsel in a correct manner his patient, because he's able to refer to the correct specialist. So the first who have to be physically active is your oncologist. Moving on to a very really current topic, this retrospective study is a small study with 36 patients with a series of limits, including the retrospective design of the study, demonstrate that people with an infection of from coronavirus disease 2019 who were physically active before having the infection had the greatest chance to have a mild disease and the lower chance to be admitted to the hospital. So in some way, again, physical activity may modulate our immune system against disease, against infectious disease and against cancer. So going to the conclusion of my speech, in the year of personalized medicine, also physical activity should be personalized in our patient. They should be supervised and gradually start with physical activity and refer to a special like sport medicine doctors or physiotherapists or the one we have available in our institution. Of course, exercise is safe and is beneficial at all stages of patient, including the ones who have an advanced disease. And I will conclude with the activity we are running, the activity we're pushing patients to. This is me, just to show that I'm physically active, but I need to admit that I didn't run this in this occasion, but I was a strong supporter. And Nordic walking, you know, the Nordic walking is a fantastic physical activity for people who suffer from breast cancer because of the reduction of lymphedema. And of course, in the bike world day, bicycling, this was our team, the Bike for Life. We collected lots of kilometers for Benefit Association. Thank you again for this invitation, the chance to discuss something which is never discussed during oncologist webinar.
Dr. Bonatti, thank you so much. And I completely agree with everything you said about the importance of having, you know, exercise interventions as part of the treatment pathway and the treatment program. And I really like your messages that it's, you know, it's never too late and you can start small and you can build up from that. So thank you so much. And hopefully we'll see more and more talks on these areas that aren't necessarily talked on so much within the care pathway. So I know time-wise, time is going against us a little bit, but Gina, please just be aware that we have been allocated about 10 to 15 more minutes. So it's really, really important that you have your time for your presentation. So please don't feel you have to rush. So it's my really great pleasure to introduce Gina Hollenbeck. Gina has been living with stage 4 lung cancer since 2015. You know, she has no family history or risk factors. Gina is a wife and a mother of 2 boys and is the president of ALK Positive Inc., which is a patient-driven group dedicated to improving the life expectancy and quality of life of those with ALK positive disease worldwide. Gina is so active within the advocacy space. She has served as a panelist and speaker for the LCT, ASCO, CURE, the GoTo Foundation, LCFA, just to name but a few, and as a reviewer on research grants for the Department of Defense. And Gina has also been featured in Time, Health, and other local media. Gina is an advocate for the early detection of lung cancer, biomarker testing, and for easier access to clinical trials. She was also recently granted one of the Patient Advocate Travel Awards to IASLC 2019, which was held in Barcelona. In addition to all of her media and speaking activities and the work she does with ALK Positive Inc., Gina and her husband Greg are also heavily involved in fundraising and community activities to educate people around them about lung cancer, the inadequate funding that lung cancer research receives, and of course, the really important message that anyone with lungs can get lung cancer. Gina is an advocate who is driven by passion to change the outcomes for all who are diagnosed with this disease. So Gina, I'll pass over to you, and thank you so very much for being here with us.
Thank you so much, Anne, for that lovely introduction. I'm going to share my slides. I hope that's okay.
Yes, perfect. Thank you so much.
Great. I am honored to be here, and honestly, just to be a part of IACAP and the Spanish Association of Lung Cancer Patients, to really include the patient voice and the patient experience is so important. I'm so thankful for Dr. Wu, for Dr. Chiarle, and all the researchers and doctors here, because what you do truly impacts patients and it really matters. So we're so thankful for the space that you're in and the things that you do. I was asked today to talk about 4 main points: how I was diagnosed, how I found out about ALK positive, what our group actually does, and how patient groups can help individuals. So I'll be talking about those key points. I'll begin talking about my diagnosis. And like Anne-Marie said, I was diagnosed in 2015. At the time, there were 2 targeted therapies. They were crizotinib and suritinib. And at the time, I was also diagnosed with a brain metastasis. And so I was automatically stage 4, which was inoperable and incurable. So it was quite scary at the time. Some major teaching hospitals in the US actually said that I only had about 10 months to live. Another hospital close to my area gave me a hospice manual as soon as they found out about the stage and that it had gone to my brain. I've endured quite a bit. I've seen the pain in my husband's eyes as I rolled back to surgery for brain surgery, the removal of my left lung, radiation, and just as recently as last week, emergency heart surgery. So I'm coming at about 6 years. I am not progression free. We're looking for the next things for patients, and all patients need to make sure that they are able to have the best care. So we're so thankful for these researchers. I'm never giving up hope. And when it comes to clinical trials and advocating for patients, I am a fierce advocate, and I feel very compelled to make sure that people know about access to biomarker testing, to targeted therapies, and also to making clinical trials much easier for patients to access. This is my lovely family. Before cancer, I was working as a nurse, and I was training for a triathlon, and I developed a non-productive cough. Like Anne-Marie said, I have absolutely no family history and no risk factors at all. So it was very surprising to my doctors because of the stigma that is associated with lung cancer. I had a very difficult time being diagnosed. At 37 years old with a cough and absolutely no history of smoking, no one really took me seriously. However, my nurse senses were spiking, and I knew that something was wrong. And so eventually I self-paid for a chest x-ray, which revealed that there was some pretty shadowy and suspicious things going on in my left lung, which I took to the ER eventually, and that's how I was diagnosed. So we can say that stigma actually prevented a timely diagnosis on my part. So it is something that we do have to make sure that we understand that anyone with lungs can get lung cancer. I was kind of asked to say, how did I find out about the ALK positive patient group? Well, we do have a Facebook support group. It is actually started by a nurse named Marita Carroll. And unfortunately, she passed away in April of 2018 of ALK positive. We can say that we're making some great strides when it comes to ALK positive, but we're still losing members. When I joined the group in 2016, we were still losing members at an exponential rate, maybe one member per week. And so as we became more aware and we worked together and we were supporting each other in this Facebook support group, about 8 of us stepped up and we said, you know, we're nervous that maybe ALK positive is too small of a population to actually make an impact in research. So we want to fund research. We were very thankful that Longevity took us under their wing and they provided a physical sponsorship for us. As a group, the very first fundraising that we did, we raised $600,000 and funded 3 research grants. One of those was actually the research to begin Dr. Awad and Dr. Kiarle's research that he was talking about with the ALK vaccine. So we're very proud to be able to be a part of that. We all worked hard to have lemonade stands and parties to be able to make sure that ALK positive research was funded and that we were making advances for our disease. As we became more organized, we created a board and bylaws. And in 2018, I was elected as the president of ALK Positive, and I have been the president of ALK Positive since. We are so excited that we have just recently created our own nonprofit status. And this was all from patients and volunteers really giving their time and energy to make sure that they progressed and that we were funding research for ALK positive cancer. This is our website, alkpositive.org. I would definitely suggest if you don't know about us, please check us out. We're doing some amazing things together as patients. One of my favorite sayings is that in nature, a snowflake alone is very fragile. It's one of nature's most fragile creations. But when we work together, we can create an avalanche and make some real change for the future. So that's what we're doing with our group. As far as coming together as an organization, just like Anne-Marie said, our mission is to improve the life expectancy and quality of life of ALK positive patients worldwide. There are some amazing things that we're able to do together. One of the things that we noticed within our group is that we had people from all over the world joining our group. Not only were they ALK positive lung cancer patients, but they were also ALK positive patients who had breast cancer driven by an ALK positive gene, sarcomas, brain cancers, and melanomas. So there were patients that were being treated with targeted therapies, and we were all sharing our experiences together. One of the things that we saw and that was very relevant was that where you live often determined if you live. So we were very thankful that one of the members of the ALK positive support group wanted to fund our second opinion program. So that was started back in 2018, where people were actually allowed to have telehealth with some of the ALK experts, the international and worldwide ALK experts. We really wanted to make sure that community doctors who didn't know much about ALK or didn't have the experience with ALK were really given the opportunity to have that second opinion with some of the ALK experts and the people who have been treating ALK for a long time. So that was our second opinion program. We're still very, very proud that any member of the support group, which is only patients and caregivers, it's a closed support group, but any member is allowed to have that free second opinion. In addition, the way that we help patients is that we have our ALK Summit. So in 2018, we had our very first ALK Summit in Kentucky. In 2019, we had a live summit in Atlanta, Georgia. we were the first lung cancer group to actually storm the halls of the American Cancer Society and show that really lung cancer was not about just smoking prevention and smoking cessation. We were survivors, and we for the most part didn't have histories of smoking. We were young people with children and families ahead of us. And so it was very exciting that we were able to work with the American Cancer Society. They worked with us to really understand our cause and who we are as lung cancer patients. We have our ALK talks on Sunday nights, where we actually invite researchers and people in the advocacy, in lung cancer advocacy and ALK advocacy, and we talk about everything ALK. And in fact, I want to give a shout out because I know that one of our ALK to ALK groups or ALK talk groups, our art therapy group, which meets weekly, is listening in right now. And so we're very thankful to have you guys listening in. And we've become such a close community. When it comes to COVID, one of the silver linings of COVID is that we all learned to use Zoom. And so we really got to find other ways to come together as a community. Another way that we're helping patients is, like I said, oftentimes where you live determines if you live. So it determines whether you're able to access clinical trials, able to access biomarker testing, and even the vital medicines and targeted therapies that are needed for ALK-positive cancer. We created our International Ambassador Group, where ambassadors from countries from all over the world work together to address the disparities. We really look at the diversity. We really want to create equity and make sure that people, no matter where you live, have access to medicines and clinical trials and treatments. The thing about the ALK positive ambassador group is that these people from all over the world who are either ALK positive patients or caregivers are working to really address the disparities and make sure that patients have access to all of these things that we need. When we're looking at it, ALK-positive cancer is not defined by the borders of country. So we believe that the science should also not be defined by countries. And so that's what our mission is to really work with industry, work with advocacy organizations to make sure that people all over the world have access to care. Oops, I'm so sorry. I wanted to thank you so much for listening. I'm going to try to make it as short as possible. I wanted you to see our amazing team. Not one person could do this. All of these volunteers give their time and energy. These are all patients and caregivers who really worked towards making ALK-positive research possible. Like I said, we funded Dr. Kiarle and Dr. Awad. Go2 physically sponsored us, and we also funded research in combination therapies the next year with Dr. Lovely and Dr. Trevor Bavona. The next year, we were actually able to fund $1.3 million and fund 2 clinical trials. One was for Dr. Awad and Dr. Kiarle's ALK vaccine, and the other one was for Dr. Dugogo Jack's SHP2 inhibitor plus lorlatinib. And so we're so excited that we were able to do that as a patient group, patients working together, bringing our resources and energy together to really make sure that we're changing the direction of research when it comes to ALK-positive cancer. I wanted to thank all of you because without you, the world would be without us. And so what you do truly matters. And I am so honored to be a part of this. Thank you so much for listening to the patient experience.
Gina, thank you so much. And I mean, the patient experience, the lived experience of this disease is the most important thing. So, I mean, you being here and being part of this meeting has been absolutely fantastic. And if there's any doubters out there that were listening to this webinar this evening, Gina, I think you've shown the importance of not only patient-centered research, but patient-driven research and the impact that that can have. So I think there shouldn't be anyone out there now that is under any doubt of that importance, and I mean, just the amazing things that can be done. Your analogy in terms of the snowflake and the avalanche, I mean, it's been underscored time and time again in all the different presentations this evening about the importance of coming together, working together for the whole community, no matter where you live, no matter your socioeconomic status. We need to do more to have more research and to really tackle the inequalities associated with this disease for our community. So we have gone over time, and unfortunately we are not going to be able to take questions. However, I just want to say a huge thanks to our audience for their huge active participation in this. And there has been a huge number of questions that have been submitted. So I'm so sorry we can't get to them here this evening. However, we are going to try and work on a way that we can answer those questions in a different means. So please bear with us as we're trying to figure that out. I just also want to say a huge thank you to all of the speakers here this evening for taking part. You've given great overview on the different parts of the pathway, the research, and talking about the areas of the pathway that aren't usually talked about, such as the pathologists, such as exercise intervention, and so on. All of these things have to be joined up and together in order to get best outcomes for people living with this disease. I'd also like to thank our collaborators, which were Takeda and Pfizer, for their support of this event this evening. It has been a great experience for LuCE to be part of this event. However, I'd like to say a massive congratulations and thank you to IACAP, who have done a huge amount of work in bringing this event together this evening with all of the speakers and all of the logistics and everything involved. So thank you so much to IACAP for that. This will be available on their YouTube channel after this event, and their ROS1 event is also available there. So I hope you can go and check that out after this. So again, absolute huge thanks. I can't say thanks enough this evening. It's been such a great event, and let's go out there and continue to work together and really make a difference for everyone impacted by this disease. So thank you all so very much and have a great evening.